dive into the Axion's innovative technology for neuron, cardiac, and stem cell research. Here is the outline of today's webinar. We will spend most of our time exploring the electrophysiology using MEA systems with neurons or cardiomyocytes. We will dive into how MEA works, the endpoint we can achieve, and some exciting application examples. For researchers working with stem cells, we will also provide a brief introduction to the live cell image system, Omni, which can track sample growth over time. Okay, we will start with the neuron assay. And so what is MEA, including what is MEA, how does MEA work, and I will introduce a few neural applications. Okay, let's begin with our MEA systems. And we have three distinct types that is called Pro, Edge, and Volt. They have the same function, just designed to meet different throughput needs. And each system is equipped with our cutting-edge Bi-core 4 chips, which provide high sensitivity and ultra low noises. And our environmental chamber is designed CO2 controlled and temperature controlled, and both the tray holder and the system door are temperature controlled, ensuring optimal conditions for cell viability and function. In addition to core electrophysiology capabilities, customers have the option to select viability and the impedance modules based on their specific research requirements. And all these systems are capable of electrical stimulation and are fully compatible with our Omni systems. The Omni will be introduced by the end of this webinar. And overall, our flexible modular design allows customers to choose the components that can best align with their research objectives. So what is MEA? MEA stands for microelectrode array, and the Axion's MEA plates are designed with a grid of electrodes embedded in the bottom of each well. As cultured neurons mature, their electrical activity can be captured by those nearby electrodes. When neurons form cohesive network and develop functional activity, the cell-cell communication or network activity can be measured simultaneously across multiple electrodes. Axion's MEA plates offer flexible throughput options, allowing to scale your assays from 6-well to 96-well formats. The MEA systems performs non-invasive electrical measurements from cultured neuron populations, eliminating the need for dyes or reporters that could interfere with your cell module or compound results. And with just the basic culture techniques, you can track viral activities over extended periods, like hours, weeks, even months, from the same population of cells. The system is incredibly user-friendly, and measurements can be taken with some simple push, and measures could be taken with the simple push of a button or scheduled in advance. There are no incubation steps or complex handling procedures required. So just making it mostly hands-free assay that streamline your workflow and enhances productivity. And getting started with Maestro couldn't be easier. All you need to do is culture your neurons in an Axion's multi-well MEA plate and load this plate into the Maestro MEA system and allow the environment chamber to automatically equilibrate. Analyze the neural activity of the neurons in the MEA plate label-free and in real time with the Axis Navigator, our software. And we are taking care of your assay so you can focus on the science. So here is some more details. of our of what we could get from the MEA assays. The neural activity on a single electrode can be quantified in various ways. Firing patterns may be random or rhythmic, or fast or slow, can occur as single spikes or clusters of spikes. Cluster of spikes, we call it bursts. So from these spikes or bursts, additional parameters can be derived, such as firing rate, burst frequency, or burst duration. So with multiple electrodes in array, you can examine coordinated activity across the entire network. So we provide the raster plots to reveal how spiking activity is organized in time and across a functionally connected network. A network burst is a coordinated cluster of spiking activity across multiple electrodes. Given the space between the electrodes, no two electrodes will detect firing from the same neuron. Therefore, coordinated activities such as network burst represents synaptic communication between neurons. After each assay, you will obtain over 70 distinct endpoints. We call those endpoints as the neuron fingerprint. The term reflects that endpoints vary depending on the differentiation stage of the neurons or the specific treatments that you applied. And with the dozens of the endpoints that you get from the MEA assays, we could divide those endpoints into three parts. Part one answers the question that are the neurons functional? And neuron function is characterized by action potentials. High activity values or high firing rate indicate that the neurons are firing action potentials frequently. And part two answers the question that are those synapses functional? Synapses are the functional connections between neurons where an action potential in one neuron can influence the likelihood of another action potential in another. And so synchrony reflects the strength of those synaptic connections, showing how likely neurons are to fire simultaneously on a millisecond timescale. And part three answers the question, is the network functional? Neuron oscillations, we call it neural oscillations, marked by alternating periods of high and low activity. And that is a hallmark of functional networks with both excitatory and inhibitory neurons. So the data recording and analysis with our Maestro MEA system and the software that are fast, easy, and powerful. And customers can use the mean firing rate heat map to get an overview of the entire plate, or dive deeper into continuous spike data, or overlay the waveform of plots at different software interfaces. And they can also export the raster plots and bar graphs under less than an hour. You don't need to be an electrophysiologist to use our system. In fact, most of our users are not. Our technology empowers you to extract incredibly powerful insights from understanding single neuron activity to analyze the plate-wide data and extracting complex information from data sets. The key point I'd like to emphasize is that from raw data recording to generating histogram of each endpoint, the entire process is highly efficient. If you are familiar with all our the functions of the software, it takes less than half an hour. So this level of efficiency in MEA data analysis is truly remarkable. After we have get a brief idea of what MEA is and what we can get from MEAs, let's move on to its applications. And here are some major application areas. Some researchers use it for a cell culture over time without interference, allowing the users to track development, the neuron development, disease progression, or chronic responses. And other researchers use it to characterize and classify cultures and their responses, identify phenotypes using multiple endpoints, and compare compounds, gene types, or cell populations. There are also customers also use it for high throughput drug screening or testing the effects of personalized medicine. And here are some disease modeling examples that we have seen from the published papers, such as ALS, Alzheimer's disease, autism, epilepsy, Williams syndrome, and so on. With our MEA plates, we offer throughput region from 6 to 96 wells. Users can maximize the throughput, the flexibility, and ease of use. The Maestro MEA system is a perfect complemented. The Maestro MEA is the perfect complement to many molecular biology assays, providing a comprehensive and efficient solution for your research needs. And so let's move on to more specific examples. Here is the data from Dr. Ann Ben at the Stanford Brigham Pre-Bio Institute. The raster plots here, we showed the raster plots of iPSC-derived neurons at week 2 and week 4 provide a clear picture of how firing activity evolves over time. Additionally, we can quantify key endpoints like mean firing rate, synchrony index, and network bursting to compare the differences in activity, synchrony, and oscillation at different stages of culture. And here is another example that the research team used Maestro MEA for autism disease modeling. And the MYT1L is a transcription factor associated with autism and is expressed in virtually all neurons throughout life. In this study, the author used genetic correction to rescue neural activity in autism model. And the data in figure A shows that the 50% knockdown of MYT1L in human iPSC-derived neurons lead to a doubling of spike firing and a tripling of simultaneous network firing activity compared to control. And in figure B, and using primary neurons from mice and replicated the data from the human neurons. They found that the increased firing and network hyperactivity scaled with MYT1L depletion. So all this data demonstrate that continuous depletion of MYT1L impairs neural function and induces an unexpected electrical hyperactivity phenotype in both neuron and mouse neurons, in both human and mouse neurons. Okay, here is another example of glioblastoma research. And the research team using magnetoencephalography, that is called MEG data. And according to the MEG data, they divide the tumor regions into two parts. The high functional connectivity regions and the low functional connectivity regions, that is called HFC region and LFC regions. And the author co-cultured the HFC and HFC or low FC tumor regions with cortical neurons. And the MEA data show that co-cultured HFC glymer cells exhibited higher, increased network frequency and synchrony, as we could see from the bar graphs here. And in figure D, we could see that after they also applied the GBP to the cultures, and the GBP, which blocks the synaptogenetic factors such as TSB. And after the 24 to 48 hours of treatment, the mean firing rate, burst, and the synchronized network burst were reduced. So this data explains the neuron dysfunction of the neurons with co-cultured with the blastoma blastoma cells. Okay, besides 2D cells, lots of users also use MEA for brain organoid research. Since MEA is non-invasive and label-free, you can continuously monitor the activity from the same organoids for many months. So here in this example, the Alzheimer's Moitris lab at UCSD generated iPSC-derived cortical organoids and tracked their activity, bursting, and network oscillations once per week for 10 months. Over time, the organoid exhibited increasingly complex activity, including higher mean firing rates, greater synchrony, an increased burst frequency, and more frequent network-wide bursts. After 4 months, we can see that higher frequency oscillations or sub-peaks emerged within larger network bursts, as illustrated here. Additionally, the network events became more temporarily variable, as evidenced by the increased coefficient of variation in inter-event intervals. And besides the brain organoid, there are also other interesting examples here. And this paper, the team, they are using the assembloids for Parkinson's disease research. So assembloids basically are just 2 or more organoids that are co-cultured and allowed to connect. In this paper, researchers combined midbrain and striatal organoids and allow them to connect to develop a module for studying Parkinson's disease. So in the top 2 figures on the right, you can see the dopaminergic neurons in red, and both in midbrain organoids and the track connecting the 2 organoids. In figure F, we can see the significant glial cell presence and marked in green. And finally, in figure G, we could see the midbrain neurons in green, extending and forming synapses. The synapses is in red in the striatal organoids. And then the team uses MEA to investigate the electrophysiology properties of the samples. They focused on the not the whole assemblies, but they focused on the area that is called the interorganoid pathways, the IOP's activity, versus the single midbrain organoids. So here is the result. The IOPs cultured on MEA began to display spontaneous low field potentials at day 14 post differentiation and reached maturity at day 25. And they also applied the blockers. They applied the GABAzen, the GABA-A receptor blocker. And so the GABAzen increased the firing rate similarly with both IOPs and midbrain organoids. Finally, they applied the the 6-hydroxy dopamine. After the treatment, the IOPs exhibited a significantly greater decrease in firing rate, and compared to midbrain organoids, indicating that IOPs are more sensitive to the 6-hydrodopamine due to the closer proximity of dopaminergic pathways to the electrical surface. So this study concludes that IOPs can serve as a more sensitive module for investigating dysfunctional dopaminergic networks in Parkinson's disease, providing a real-time functional readouts of research. And this is the last example of neuron axis. The team is using acute brain slice for on MEA. So this study, they tested the neuron activity in the striatum of 10-month-old APPPS1 mice using MEAs. The samples they used were brain slices, so the team and also applied high current stimulation to the samples. And the gene they were studied is MECP2. It's a methyl CpG binding protein 2, which is an important epigenetic regulatory protein encoded by the MECP2 gene. And it plays several key roles in the nerve systems, including the maintenance of neural function. And the MEA data show that knocking down MECP2 expression in the striatum rescued altered neural activity in those symptomatic mice. The high current stimulation showed significantly reduced firing in APPPS1 mouse, which was restored in the MECP2 knockdown group. And on the top right, the heat map images also indicated rescued firing patterns. So overall, the MECP2 knockdown in the striatum can rescue defective neuron phenotypes in this Alzheimer's disease model. Okay, that's all for the first part, the neuron axis of MEA. So now let's move on to the cardiac axis with MEA. We will also talk about how does it work to detect the cardiac electrophysiology signals, the 4 measures of cardiac function, and I will also give a few examples. So if we culture the cells similarly as neurons, when cardiomyocytes are cultured on those electrodes, we can monitor the emergence of cardiac activity and observe the formation and maturation of the syncytium. This allows us to quantify the properties of cardiac beats and classify the behavior of the cells. And getting started, cardiac axis with our MEA system is as easy as we can do with neurons. Just culture your cells in an MEA plate and load the plate in the system and record and analyze the data with our software Axis Navigator. So with our Maestro platform, you can record 4 key measures of functional cardiac performance. They are field potential, propagation, action potential, and contractility. So I will introduce each of the 4 key measures. So we will start with field potential. Sometimes we also just call it FP. So when cardiomyocytes are cultured in MEA plate, spontaneous action potential from a group of cells can be detected as fluctuations in the extracellular field potential at the engine recording electrode. The recorded cardiac field potential signal arises from the propagation of the extracellular cardiac action potential across the cell monolayer relative to the recording electrode. So in this respect, the recorded field potential signal is analogous to the recorded ECG signal that measures changing voltage on the body surface induced by currents that propagates across the heart. And the field potential waveforms here, the field potential waveforms comprises a sharp transient spike associated with sodium influx and membrane depolarization, followed by a shallower ramp during calcium efflux, ending with a repolarization wave that corresponds to the potassium efflux. And our system can provide the parameters that our system can provide, including the depolarization, the amplitude of the amplitude of this beat, and repolarization FPD, we call it. It is field potential duration and the beat timing. And we could also use our waveform to make the judgments of the irregular beating or arrhythmia. And a prime example of the Maestro MEA exceptional performance is demonstrated through the CEPA initiative. The CEPA is short for Comprehensive In Vitro Arrhythmia Assay, which aims to update cardiac safety testing paradigms to more accurately evaluate the arrhythmogenic risk of new compounds. So in the CEPA pilot assay, 6 core sites use the Maestro for their MEA studies with 2 different cell types, Core4U and iCell, and 3 compounds with known mechanisms. And here is the example raw field potential waveforms. So the black line are the baseline waveforms, and the orange and the blue waveforms are the treatments or the dosing waveforms. The Maestro reliably detected the field potential duration. We can say that they detect the field potential duration prolongation for the hERG potassium channel blockers, and the FPD reduction for calcium channel blocker, and spike amplitude reduction for sodium blockers across sites and cell types. And here is the summary of the data of the pilot studies. And the Maestro, we could say that the data demonstrated the high reliability across replicate sites and the cell types, while we accurately measure the changes in depolarization, repolarization, and detecting arrhythmias. And all those data have been published on the biomaterials in 2018. Remember, this is a multi-site blinded study. We are proud that the data from Axion MEA systems proved to be the most reliable and most accurate with lowest viabilities. And here is another example using MEA for 3D sample electrophysiology. And in this study, scientists from Korea have created self-organized heart organoids, validated the organoid's electrophysiological function with Maestro MEA. The team also optimized the conditions for the seeding of the organoids by reducing the volume of the media to make sure that the organoids have a good attachment with the electrodes. Then the organoids activity were recorded at day 13 and day 19. And the changing of the beating time and amplitude of the field potential are similar to the increase. From the data, they found that the changing of the beating time and the amplitude of field potential are similar to the increase in the amplitude of the action potential in reports reviewed by maturation of cardiomyocytes. And they also observed a tendency similar to the patterns of in vivo heart rate that occurs during maturation of fetal heart to adult heart. And in the paper, the team also applied several compounds to the organoids. So if you have interest in this research, you can go to find the paper for more details. And let's move on to the second key measure that is called propagation. And when cardiomyocytes are cultured in the MEA plates, we know that there are multiple electrodes in each well. So each beat is initiated in one part of the culture and propagates across the monolayer. So the measurement of cardiac beat propagation and conduction velocity is only possible when an array-based with an array-based approach. So the Maestro MEA platform be equipped with from 8 to 64 recording sites per well, allows for the detailed measurement of changes in propagation patterns and conduction velocity. So this capacity is particularly useful for pharmacological integration and during the differentiation of the cardiomyocytes. And the Maestro Edge can give the key parameters of propagation, such as the conduction velocity. And we can also use the heat map to display the change of the conduction velocity or the propagation pattern. Here is an example. An anti-cancer drug is added to the iPSC-derived cardiomyocytes and caused a disruption of propagation pattern, and thus a decrease in propagation consistency from bit to bit. And here is another example that detects both the field potential and the propagation of cardiomyocytes for myocardial infarction research. And in this paper, the team used the polydopamine nanoparticles served as a bioactive agent to treat the cardiomyocytes. And the MEA data showed that the treatment caused a concentration-dependent decrease in beat period and FPD, and an increase in spike amplitude, we can see here, and suggesting that the treatment with the nanoparticles enhances the depolarization of the cells. Furthermore, a similar response was observed in conduction velocity across the cells. It's also worth mentioning that in figure D, and the treatment of the nanoparticles also enhanced the beat propagation consistency and conductance. In the figure E, we could see a significant decline in the beat period irregularity was observed after the treatment. So taken together, these findings suggest that the nanoparticles exhibited strong bioactivity on conductive cells such as the iPSC-derived cardiomyocytes, and can modulate electrophysiological parameters influencing the function of those cells. Okay, let's move on to the third measure. We call it LEAP. LEAP is short for local extracellular action potential. And the field, as we have discussed, the field potential signal is detected based on the intimate contact between the cardiomyocyte syncytium and the electrode. We have developed a patented technique to enhance the coupling between the cardiomyocytes and the electrode. So as a result, and after the induction, and the electrodes can detect a larger signal, which we refer to it as a local extracellular action potential. Because the waveforms of the field potential and the leap is quite different, but the waveforms of the leap is closely similar to the transmembrane voltage of an action potential. That's why we call it local extracellular action potential. Sometimes we just call it action potential on NEA. And the key parameters of the leap that we could provide, including the depolarization, the rise time, and the repolarization, such as APD 30, 50, and 90, triangulation, and irregular beating or arrhythmia. And the advantage of the leap are highlighted by a few case studies in pharmacology. Here we illustrate the change in the leap signal in response to a block of calcium and potassium currents. So here each waveform was acquired from an individual wheel and is referred to the vehicle control. So the black line is the vehicle control. And as expected, the concentrations of nifedipine, which L-type calcium channel blocker, progressively shortened the duration of the leap signal. And so here is another example. The increasing concentrations of E4031, the blocker of the hERG potassium channel, and first prolonged the repolarization and then generated repolarization irregularities. Finally, we apply the multi-ion channel, the verapamil. It both block the calcium and potassium currents. And so the treatment shortened the leap signal and eliminated the plateau phase of the action potential. Okay, that's the third one. Let's move on to the fourth, the contractility. When cardiomyocytes are cultured on MEAs, they form a spontaneously beating syncytium. As the cardiomyocytes mechanically contact and relax over an electrode, the shape change is detected as an increase and decrease in impedance. So the array of the electrode can detect the regions that are contracting while other regions are being stressed. This pattern can be represented as a contractility map, where the relative size of each orange circle I ONIFL is a plug-and-play live cell imaging system designed for continuous multi-well image in your incubator. So it's totally different from the MEA system that we have discussed. It's a live cell imager in your incubator. So the whole complex, with this system, the whole complex biological process of the cell growth or cell dying is recorded through time-lapse imaging. You can get the whole story. It allows you to pinpoint the time important events in the progression of the cell culture from experiment to experiment. Researchers can use flux dishes 6 to 384 well plates for image acquisition. It's compatible with the high throughput analysis. With automatic bright field and fluorescent imaging, customers can study complex biological processes with easy. And all the scannings are uploaded to the cloud portal automatically. So the integrated multiple analysis module allows user to inspect their cells anywhere, anytime. And so here is a few details of the OmniFL. There is an arm here, and the arm can move in horizontal direction. And there is a light art with LED, and the arm is 55 millimeter. So any plate or vessel that is transparent and face the arm can be used. And in the corner, there is actually, we didn't, maybe it's not as clear as you can see, but there is, in the corner, there is a L-shaped bracket. So you can align your plate or vessel right in the corner every time. So it's consistent. And it has a 6.4 megapixel CMOS, 10 times of objective with 20 times of digital zoom. And here is the excitation emission waveform length of green and red fluorescence module. So the results that you get can be downloaded as JPG, TIFF, MP4 videos, or Excel files of the graphs. And so I'd like to highlight the one important feature of the Omni products, that is all the images are analyzed on the cloud portal. And there are many benefits of the cloud, so you save money and you don't have to buy server or external hard drives for data analysis. Just a laptop will make sense. And the cloud analyzing is faster to get results during experiment and also faster than local processing. And with unlimited storage and automatic backups, you never have to worry about losing data. You can access your data with phone, PC, no need to go to the lab to monitor your experiment. And each person have their own account. So nobody can read your data if you don't share with them. And you always use the latest algorithm and software with the cloud. And we also offer 6 different software modules available to meet the customer's SEs. We will they are Confluence module. The Confluence module help you to track the cell growth or cell death in your experiments in bright field Confluence. And there is also the fluorescence module. And so we could get the fluorescence confluency, the intensity, and object detection. And there's also a stretch assay. We could investigate the progression of wood closure over time. And there is also the a clonogenic assay, a clonogenic module, and an iPSC module. All those 2 modules we can help us to track the clone formation and to study the effects of the cytotoxic or immunotherapy agents. And there is also an organoid analysis module available to monitoring the growth of the iPSC-derived organoids or other organoids over time. And so with our OmniFL, there are lots of applications that are available, and like cell culture growth and oncology and immuno-oncology, and the migration of wood killing, cloning formation, or for the drug compound screening, cytotoxicity, or the spheroid or organoid formation. And there is also a few customers to use it for the microfluidic chips to track the cell growth or cell movement over time. So that's all for the Omni. But last but not least, I'm excited to introduce that our Connect Lab. So Connect Lab, it can streamline your MEAs with the OmniFL. So with those 2 systems, all you need to do is just use OmniFL to scan your MEA plates, and then the images can be uploaded to the cloud. And with our software, the Axis Navigator, so there is a video here I will show you. So with the Axis Navigator, which is the can automatically download the images scanned by the Omni, and according to the barcode of the MEA plate. And so after the downloading, you can monitor your cells' electrophysiology, their firing activity, and also as well as the cell morphology, or does the cell have a good attachment to the electrode? So you can monitor the cell culture quality over time, or you can use it to optimize the MEA assays with imaging. So the Connect Lab function can help you to get a more comprehensive view of your samples with side-by-side imaging and MEA data. Okay, that's all for today's webinar. Thank you for your attention.